Welcome to this video on challenges, strategies, and regulatory considerations for the design of rare disease clinical trials. FDA understands that patient time and effort is very valuable. Therefore, we strive to work with all stakeholders to maximize clinical trial efficiency and the likelihood of interpretable results. Scientific issues such as small heterogeneous populations, varying time courses and manifestations of disease, and natural history that is not well understood make rare disease drug development uniquely challenging. Understanding the urgent need of rare diseases which lack approved treatments, FDA recognizes that different trial designs may be appropriate in different disease settings. For FDA to approve a drug, the law requires that a drug's effectiveness be demonstrated by substantial evidence. FDA has interpreted this substantial evidence requirement as generally requiring 2 adequate and well-controlled clinical investigations. Characteristics of an adequate and well-controlled clinical investigation include a design that permits a valid comparison with a control. Methods of assigning participants to treatment and control groups are intended to assure comparability of the groups, and methods of assessment of participants' responses that are well-defined and reliable. A complete list of characteristics is displayed. The law also permits FDA to determine, based on relevant science, that data from one adequate and well-controlled clinical investigation plus confirmatory evidence are sufficient to demonstrate substantial evidence of effectiveness. Depending on the circumstance, types of confirmatory evidence include clinical evidence from a related indication, mechanistic or pharmacodynamic evidence, evidence from a relevant animal model, evidence from other members of the same pharmacological class, natural history evidence, real-world data and evidence, and evidence from expanded access use of an investigational drug. The one adequate and well-controlled clinical investigation plus confirmatory evidence approach is frequently used in rare disease drug development. FDA considers a number of factors when determining if a single adequate and well-controlled trial plus confirmatory evidence meets the substantial evidence standard. These include the persuasiveness of the single trial, the robustness of the confirmatory evidence, the seriousness of the disease, the unmet medical need, the size of the patient population, and the practicality and ethics of conducting more than one trial. There are also other important considerations which impact the approval decision, such as whether a drug is safe for its intended use. These considerations are not discussed in this presentation. Rare disease drug development is not one size fits all. Randomized, double-blinded, concurrently controlled trials are usually regarded as the gold standard, since these trials can provide more reliable evidence of a treatment effect compared to other designs. However, FDA recognizes that certain design features may be appropriate when the disease is rare with a high unmet need. For example, sponsors may assign more participants to receive the test drug than the control, or conduct a dose comparison design to increase the number of participants receiving the investigational drug product. In some circumstances, sponsors may also use crossover, randomized withdrawal, or randomized delayed start designs, or a single-arm trial with an external control may be used. However, the suitability of using an external control is informed by several factors and warrants a case-by-case assessment. Adaptive clinical trial designs present another opportunity to efficiently study rare diseases. Adaptive designs allow for prospectively planned adjustments based on information unavailable at the start of the trial and can more efficiently gather data in a single trial. For example, in a trial studying a drug intended to slow functional decline in a chronic and slowly progressive degenerative disease, there may be uncertainty in the time needed to demonstrate a difference between the control group and the experimental drug group. An adaptive design may incorporate a pre-specified time point to evaluate the effect of the test drug. This strategy can avoid the risk of stopping a controlled trial too soon, before a treatment effect can be reliably demonstrated. More information on adaptive trial designs can be found in the FDA's guidance, Adaptive Designs for Clinical Trials of Drugs and Biologics. Potential advantages of adaptive designs include statistical efficiency, ethical considerations such as the ability to stop a trial early if it becomes clear that the trial is unlikely to demonstrate effectiveness, thereby reducing the number of participants exposed to an ineffective treatment. Improved understanding of drug effects, increased acceptability to stakeholders, for example, response adaptive randomization, can increase the probability that participants will be assigned to the more effective treatment. Another important feature of rare disease trial design is dose selection, and generally, the effects of more than one dose should be studied. Given the limited number of patients, multiple data sources should be used to select the dose for safety and efficacy trials. Crossover or seamless phase 2-3 designs, including those with parallel groups, may be considered for dose optimization and efficacy assessment in the same trial. And integrated analyses of all data from other clinical studies or model-informed approaches can also aid in dose selection. Rare disease drug development is most successful when it is a collaborative effort. The best approach is to work with FDA early in the process, before designing efficacy trials, so that we can work together towards the goal of developing new therapies in the most efficient manner. Meetings between a sponsor and the agency are frequently useful in resolving questions and issues raised during the course of drug development. For earlier stages of development, we offer several types of meetings, including CPIM, Interact, and Pre-IND. Additional information, including relevant guidance documents, can be found on the ARC website. This video was created as part of CDER's Accelerating Rare Disease Cures program. For more info on relevant guidance documents and the ARC program, please visit www.fda.gov/drugs/CDER-ARC.